Good morning, students. In this lecture, we will cover about the membrane separation. Firstly, if we talk about the membrane separation, this separation process, it is extensively used in the chemical process industry, and their use in food industry is now becoming the more common. So, uh, as this membrane separation process is having a wide application in the food industry. So firstly, I would like to tell you about what is actually membrane separation. So in case of the membrane separation, a membrane is used that may be a plant based membrane or a animal based membrane. This membrane is used, and by using this membrane the separation of the certain compounds is done. Example you can take, actually that is not actually a membrane, but it is kind of the separation process. For example, the sieve which we use at our home for pouring the tea, for removing the certain things from the floor. So that sieve, you can consider that sieve as a membrane, and that separation process, it is known as the purification or the separation process by using the membrane. So whatever the thing which retain over the surface, that is known as the retentate, and the thing which get passed through that membrane, that is known as the permeate. Example I would like to take for the tea. When we prepare the tea, we put tea leaves into that. So after preparation of the tea, when finally we pour the tea, what happened? The water or the tea, it gets separated in the cup, and the other you can say the waste material, the tea leaves, they all retain over the surface. So these tea leaves and these things are known as the retentate, and the tea which you will drink, that is known as the permeate. So this is the process of the separation. Now here we will talk about the membrane separation. So as it is written here, some of the food related application include the purification of water and the concentration and clarification of juices, milk product, alcoholic beverages and wastewater. These are the certain cases where you go for the membrane separation process. Now, in a membrane separation system, a fluid containing two or more components is in contact with the membrane that permits selected component, for example, water in the fluid, to permeate more readily than other component. So same thing which I told you, that the thing which get passed through the membrane, that is known as the permeate, and which retain over the surface, that is known as the retentate. So there are several factors on which this membrane separation depend. So firstly, the physical and chemical nature of the membrane. What is the physical and chemical nature of the membrane? So what includes under that? For example, what is the pore size, what is the pore size distribution, and affect the separation. These are the factors which affect the separation process. Now if you will see the figure given in the next slide, so I'm coming on that slide. So this is that slide. Here I have shown one figure, and it is defining the different microfiltration, ultrafiltration and reverse osmosis. So what is the meaning of these things? Firstly, microfiltration. In case of the microfiltration, the pore size is quite you can say quite large, and it allow the passes of the certain component, but it suspends some particle, that is known as the suspension particle. So microfiltration is used in that condition. Then ultrafiltration, in case of the ultrafiltration, the pore size of the membrane is smaller than the microfiltration, and it also hold the macromolecules, you can say, which are having the large size. Now come to the next part, that is the reverse osmosis. In case of the reverse osmosis, it basically contain the salt and sugar, whatever the present in the case of the water, or ions, you can say, which are present in the water, it will retain over the membrane, and further water will be passed. So basically, what is the major difference between the microfiltration, ultrafiltration and reverse osmosis? The major difference is the pore size of the membrane. In case of the microfiltration, pore size is large. In case of the ultrafiltration, pore size is smaller than the microfiltration, and in reverse osmosis it is very small. So now come to the first slide. It was written that as shown in figure 11.1, the membrane is a reverse osmosis system which allow water to permeate, whereas salt and sugar are rejected. So in case of the reverse osmosis process, salt and sugar, they remain over the surface. Now ultrafiltration membrane are useful in fractionating component by rejecting macromolecule, the same thing which I discussed. In microfiltration, the membrane separate the suspended particles. Now, permeation of the selected component is a result of the driving force. For dialysis, the concentration difference across the membrane is the driving force, whereas in case of the reverse osmosis, ultrafiltration and microfiltration, hydrostatic pressure is the key driving force. Now, what is the meaning of this driving force? Driving force means the certain force is required in order to pass the liquid from that membrane. So in some cases, here it is written that permeation of the selected component is a result of driving force. For dialysis, the concentration difference across the membrane is the driving force. Concentration difference. What is the meaning of the concentration difference? The meaning of concentration difference is if you are using a membrane and at the one side of the membrane there is large concentration of the solute, and over the second hand there is less concentration of the solute. Or similarly, you can consider in case of the water, if you are having a membrane and at one side of the membrane there is large concentration of water, and at the second hand there is a less concentration of water. So in these condition, always the flow will be from higher concentration to the lower concentration. So this concentration difference, it basically act as a driving force when we talk about the dialysis process. Now, but in this case, when we talk about the reverse osmosis, ultrafiltration and microfiltration system, hydrostatic pressure is applied, or it is the key driving force. Means we apply our liquid with some kind of force, and that is known as the hydrostatic pressure. Now come to the electrodialysis system. So electrodialysis system, it is the separation process in an electrodialysis system is based on the selective movement of ions in the solution. The membrane in this system derive their selectivity from the ions that are allowed to permeate through them. Selected ion are moved from the water as they pass through the semi-permeable membrane, and these membrane do not permit permeation of water. When we talk about the electrodialysis system, in case of the electrodialysis system, only the certain ion are allowed to pass. It is also impermeable permissible to the water, means water is also not allowed to pass through this membrane. So how actually this work? As the name is suggesting, electrodialysis system. Electrodialysis system means we are using the electricity, or we are using some electric power or electric current in this case. So what happened, whatever the membrane we used, we bind this membrane with the certain ions. So these may be the positive ions, these may be the negative ion, and on the basis of these ion there is the separation of the particular ion from the membrane. Now if a polymeric chain in a membrane have fixed negative charge, it will repulse an ion that tries to enter the membrane. This is shown in the figure. In the next figure it is shown. For example, a negative charged chain attracts cation and allows them to move through. So what will happen? For example, we are having the membrane and we have binded this membrane with the Na positive ions. So we have binded with the Na positive. So now what will happen? Whatever the positive charge will come in contact with that membrane, it will repel. As you must be knowing the electrostatic law, that the similar charged repel each other and opposite charge attract each other. So if negative ion will be present there, it will repel any anion. If it will come in contact with the membrane, it will repel the anion, but it will attract the cation. If any positive, if any in case of if the membrane is binded with the negative charge, so if any positive charge will come in contact with that, so it will attract towards each other and it will allow to pass through the membrane. So this is the system of the electrodialysis. Now, the electrodialysis system uses an electric current to transfer ion through the membrane. The membrane have fixed ionic group that are chemically bound to the structure of the membrane. As I told you, in this condition, your membrane, it is binded with the certain ions. The electrodialysis system involves an array of membrane altering between anion exchange and cation exchange membrane. So what is the meaning of that? It is containing the array of membrane, means there are number of membranes are present, and these number of membranes, they possess different charges. Some are binded with the negative charge, some are binded with the positive charge, so that the different ion can get attract or passed through the different membranes. Now in between the membranes are small compartments that contain the solution, and electrode are used to apply the electric charge to both sides of the membrane assembly. Depending upon the fixed charge of the polymeric chain in the membrane, only the compatible ions are allowed through the membrane. The similar thing which I explained. So this is the diagram is shown here. So if we see that the above membrane, this anion selective membrane, it is binded with the positive charge, so it will just allow the passage of the negative ion. So in the second condition, it is binded with the K positive and Na positive, so it will just allow the passage of the negative. So this is the case. Now this process is used for the desalting process. Desalting process means to remove the salt from the water. Now the energy consumption for the electrodialysis process is given by energy consumption. How much energy will be used during this process? It will be depending upon the equation is given, E equals to I square nRC T. What is E? E is the energy consumption, how much energy will be required for the process. I is the electric current, means what is the power of the current you are providing. N is the number of the cells, how many membranes are there in the cells. RC is the resistance of the cell, what is the resistance of the cell, and T is the time, for how much time you are passing it. So now the electric current, this is the formula for calculating the electric current. The electric current can be calculated that ZFM delta C upon U. Z is the electrochemical valency, F it is the Faraday constant. This is the formula for calculating the electric current. You have studied this formula in your 12th standard. So F it is the Faraday constant, M is the feed solution flow rate by which you are feeding the solution, delta C is the concentration difference, and U is the current utilization factor. Now what you have to do, you have to place this value of I in the equation, this E equals to I square and RCT, and you will get the following equation. So this is your electrodialysis system.